Hi, everyone. If you have been tuning in to the Big Give all day, bless you, because we have had a packed schedule. I have loved talking with everybody that we've had join us, and I'm really excited to have Dr. James Fink join us. He was one of our first interviews on our original Big Give. It's been really great to watch your research grow over the years as you've come on and present for us, and I'd love to talk about it. You came to the Alliance as a research fellow, correct?
Yeah, that's right. So I think in 2015, myself and Kevin Hope were the first fellows through the Duke 15Q Alliance. Was sort of wear that as a badge of honor. It's something I'm extremely proud of in my research career. And yeah, being part of the first Big Give and still coming here now, a couple years later, is very special to me.
I love that. I love that. What's the transition been like? So catch everybody up on where have you been since 2015 when you first started with us?
Yeah, so I can talk a little bit about the fellowship because that relates, I think, directly to what I'm doing now. So currently I'm at a biotechnology company in Cambridge, Massachusetts. We're actually working on a therapeutic reagent for Dup 15Q, so it's sort of important for our company as well. And it relates sort of the work that we do to the work I was doing in graduate school as part of the fellowship funded by the Dup 15Q Alliance. So at the time, I was working with Dr. Eric Levine and Dr. Stormy Chamberlain, who I know also spoke as part of this event, and working with induced pluripotent stem cells. So this is an important point because patient cells, especially related to the brain, are really hard to access. Often we have to use post-mortem tissue, which can be useful for studying the endpoints of syndromes and disease, but it's really difficult to understand the beginnings and the happenings while it's going on. And of course, we can't actually access brain tissue from living, functioning human beings. So stem cells, traditionally you can get them from various sources. Now we can take them from blood, skin, even different epithelial cells, and using some cool tricks, we can turn them into stem cells. And then those stem cells can become any cell in the body. And of course, with Dup 15Q, we're very interested in the brain. And so at the time with Stormy and Eric, we were using these brain cells derived from patient stem cells to study cellular function of Dup 15Q. So neurons, which are the other name for brain cells, fire electrical signals. And we are trying to understand how those electrical signals were different between Dup 15Q syndrome cells and control cells. And that uncovered a variety of really important findings. And again, that work was all funded by Dup 15Q. Now, the technology I was using to perform that work in Eric's lab was taking wires hooked up to an amplifier and using this instrument to take a microscopic needle towards a cell, and then using that needle to get very, very close and onto a cell. And then that allows you to listen in on how the cell was signaling. And that's pretty laborious. So, I would say probably while this conversation happens today, we could do something like 4 or 5 cells. To understand subtle differences in the brain, you need many thousands of cells. There are some ways we were doing this in Eric's lab to get better proxies in a faster way. But that paper was published, I think, this year. We can talk a bit about that. So 2015 was when the fellowship started. The research was published now in 2022. And that's because it takes so long to collect those measurements. So in terms of what I'm doing now, QState Biosciences is really interesting in the sense that it has a unique technology that allows you to observe neurons using movies rather than these little microscopic needles. And that, as you can imagine, watching a movie, you can watch hundreds of things happening at once. And so this allows us to collect data from similar numbers of cells, but in a much shorter period of time. And so I think we're taking the work that was done as part of the fellowship, these findings from Dup 15Q cells, and using that to test therapeutics to find things that actually correct that behavior. So it directly ties into the work we are doing in Eric and Stormy's lab. And in fact, we have those cells at QState Biosciences. and so we're using them to help develop a therapeutic.
It's absolutely amazing when I think of what what people do on a day-to-day basis to combat and look for therapies. I mean, it just blows my mind as you're talking. I'm going, oh my gosh, like this is just, it's mind blowing. I guess not to get too far ahead of us, but I also want to ask you, what are you hopeful for in doing this? Like, is there a certain, if a family's watching and they're like, oh, I wonder if that therapeutic will help me in this way or help our child in this way. What kind of in your career with this are you hopeful to see happen in therapeutics in the next, let's say 10 years even?
Yeah, I mean, I'm hoping that this is something that can sort of benefit patients on that timeline. You know,
Yeah.
the really amazing thing about the transition is, you know, applying for the fellowship in 2015 as a student, doing bench research, which of course is extremely important because that work went on to sort of help push some of these ideas now in this therapeutic development. But when you're a student and you're working at a university, you're pretty far from the patients and families. I had a little bit different experience because, of course, because I was going to the conferences and meeting you all, getting to meet the patients and their families, and again, that was very special. So, you know, I had some proximity to that. But many people working in the labs don't have that firsthand experience. It's very different now being at a company where you're actively developing drugs for patients. You're sort of very close to that part of the process, and that's extremely exciting in that way. So that's what I'm hopeful for. I think if you had asked me that question in 2015, my hope would have been sort of what's come true so far, which is my work and the work that was funded by Deep15Q Alliance may someday be used by someone to help patients. And now I think we're much closer to that. And the place that I am now at QState Biosciences, we're even closer than a few years ago when we were just doing the basic research.
Yeah. Yeah. Go ahead, Naka. Did you have something?
Yeah, I just wanted to talk a little bit about the published research that you did through, and then looking at it. Now I remember, and I'm going to post the past videos where you had shared like the slide with the cells and the fire and things like that, because I think it's incredibly interesting. And especially that you were doing it like one cell at a time at that point. But to put into lay terms and just looking at the results that was posted from your abstract, it says that you were able to identify that Deep15Q neurons or brain cells are hyperexcitable and create seizure susceptibility. Like, is that what you're finding in controls? And that knowing that you'd be able to find like a targeted treatment to like get them to find some chill?
Yeah, so one of the approaches you can take when you're sort of trying to find a therapeutic is you try and establish differences between the disease state you're trying to study and the healthy state. And so, you know, using patient neurons, which again are enabled by this really groundbreaking technology, which has already won the Nobel Prize in a very short period of time, it allows you to take cells that have the same genetics as the patients. It's not a mouse model, which, you know, to be clear, those have their place in research, and there are mouse models that are trying to get at Deep15Q. But having cells that have come from the patients themselves, the duplications are exactly the duplications the patients have. That allows you then to look at what really is different between those cells and the controls. Now, it's really tough to link a cell who's this microscopic sort of unitary thing to seizures or language or muscle tone. But one of the interesting things about this hyperexcitable phenotype you mentioned, Naka, which is that you can imagine, we know from seizures in the brain when you're measuring with EEGs, which many of you families are familiar with, is that a lot of that is because the brain is very active. And that activity comes from these electrical signals I was talking about. They're called action potentials. And those are what you study when you go one cell at a time with this wire. You're measuring action potentials. And so looking at Deep15Q cells that have many more action potentials per unit of time than healthy controls, it's one of the easier things to imagine links to something like seizures. Again, it's really hard to make that connection, but it's not that hard to imagine that a cell in a dish in the lab that's very, very electrically active may relate to EEGs and seizure activity when the brain itself is very, very active. The animal models are really important for linking those things because there the animals can have seizures and you can measure their cells and see if something similar is happening. But certainly the human cells pinpoint, again, related to directly the genetics of the patients, what might be happening in the cells from those kids. And then with that in hand, you can start to explore different molecules that may rescue or correct that excessive electrical activity. And just to link it back to some of the words you're using, we call this hyperexcitability, and that's because the technical term in brain cells, when they are firing these electrical signals, we call them excitable. So it means that there are certain cells in the body, and brain cells are one of them, where they actually are electrically active. And so, if they're hyperexcitable, that's just indicating that they have much more, they're prone to exhibiting higher electrical activity than the base state.
So when you had sorry, Carrie,
No.
when you had, you said you're using skin cells or whatnot to create, these mini brains. Did you have different types of D15Q variants? Did you have like an IDIC and interstitial, or was it just strictly an IDIC? What did that look like?
Yeah, so at that time it was 4 different isodicentric or IDIC patient lines. We are very interested in the interstitials, but as many of you know, the sort of the presentation there can be less severe. And so you want to start with the sort of most severe case in case you don't want to miss any differences. And you could imagine that depending on the state of the cells from the interstitial, you might miss something. So we started with, what does the most severe cases look like? I will give a plug for Eric and his student Marwa, who I think just graduated recently. She's been at the D15Q conferences as well. They just published some work looking at the interstitials and also looking at isogenic lines, and they see very, very similar types of behavior. So I want to be clear to everyone listening that this does seem to be true, not just for IDICs, although that's the work I was doing at first.
And then where did you get the skin cells? Is there a place still taking them?
Yeah, so this is a great point because I actually was speaking to Vanessa recently. Of course, you're all familiar with Vanessa and being involved with the CMV Commission this past summer. I know the Alliance was very active in collecting samples from patients, which, again, through blood, skin biopsies, things like that, those can be used to generate these types of cells. It was less the case when I was a student. So coming by patient samples was much harder. We worked with cells in collaboration with Stormy Chamberlain. So they came from a variety of sources. And that's why we only had 4. So again, the work I was doing in that paper, it's just 4 lines. Mm-hmm. You really want to work with many, many more than that if you can. And the work that the Alliance is doing and the CMV Commission, that makes that type of stuff possible. So now, not just for D15Q, but for many of these neurodevelopmental disorders and the alliances and consortiums associated with them, they collect and bank big batches of tissues, and they come from the familial siblings or parents and the patient themselves. And that actually makes the research even better. So at the time, a very different place than we are today. They came from a few different sources and in collaboration with Stormy Chamberlain, but now there are much larger patient and familial samples available.
Now, if a family wants to get involved in donation or sample donations, is that something, I know we will certainly talk about doing that with our next conference, but is that something that someone can do if they live close to where your lab is? Or can you talk a little bit about that?
Yeah. So yes, oftentimes the samples are collected during conferences. QState does have infrastructure in place and IRBs to be able to collect patient samples for certain studies. So it's not something that we're doing right now for D15Q, again, because there's a lot of samples available, but that is something that we have the infrastructure to do.
Okay. I'm looking to see if we have questions. We've got quite a few people watching right now and interest and just how interesting this is. That's right. We're waving like, that's great. That's great. what are you, we talked a little bit about what you'd love to see happen. Is there anything in particular that for families who are newly diagnosed, I've today my theme has been hope in my heart today of just like giving families hope. Is there anything that you see coming down the pipe that would that brings that hope? Like, I mean, obviously you just talked about the advancements since 2015. That's remarkable. Should parents expect that, they can see some major changes in therapeutics in the next, like we said, 10 years? I mean, is that really that hope there?
Yes, I think that there's lots of reason to be hopeful. Again, I want to reiterate that for QState Biosciences, Dup15q is front and center for us. That is our lead therapeutic program. So we really, really care about working on Dup15q. And I've been involved with the Alliance for a long time, and I don't know if that's always the stance that companies have taken. It's not to say that people didn't think that Dup15q is important, but of course it was less known, and we've been sort of come a long way since then. And so people are focusing on this now. And that our goal is to develop a therapeutic for Dup15q. So I think there's lots of reason to be hopeful. It's not just us. I know there's other people out there working on this type of stuff. There's still lots of research. The conference has gotten bigger in terms of researchers going there. People know more about Dup15q. It's not just Angelman anymore. So I think there's lots of reasons to be hopeful on that timeline. So even today, I'm very hopeful.
I love that. And we've been really talking to the community about the importance of putting their information into and registering in our database, in our ladder database. As a researcher, how important, I mean, you've kind of touched on it just now, but how important is that for them to just register and get some data in there? I mean, talk about that.
I think that's incredibly important. Not just the cells, of course, that's really important for research. I think understanding the different symptoms that the patients have, that's also really important for matching the cells to certain patient indications. I know it's also very important to understand these things for clinical trial development. One of the challenges with lots of neurodevelopmental disorders is really trying to understand what is the readout. What is it we're trying to correct in the patients? And that can have really important indications or implications, I should say, for the outcome of those trials. So the more data you have about the patient groups that you're trying to study, that really, really helps. So I really want to encourage people to sort of be involved in those things, be open to them, because it is really crucial.
I love that you said that. And I actually watched Nako's face for a second, kind of go, yeah, that makes sense, because we often tell people too, don't just put it in their information in when things are not going well. Like, we need to know, like, is there a trajectory during a certain age that we're finding that from 12 to 14, X happens and this. So we encourage people we want this data along the entire growth curve, if you will. So I love that. Such a good reminder. And just because we are really heavily asking people to do this, we're going to continue to put on our Facebook page, laddertotreatment.org, and for people to sign up if you haven't yet, because we really, really want to make sure we get those numbers into the system so that so that pharma sees and they're interested. And like you said, there is more interest now. And speaking of that, can you share then why QState is interested? Did that come from your work? Is that because the numbers are growing? Can you talk a little bit about, as far as behind the scenes, what makes a company interested in a rare disease? And you don't have to specifically speak about your company, because that may cross some muddy lines, but is there anything you can educate us on with that?
Yeah. So, genetics is part of it. That's one. Two is, of course, Dup15q is related to the brain, central nervous system. And the technology at QState and our entire system that we've built to understand disease is really well suited for central nervous system disorders. And then, given that there are these hyperexcitable patient cells from the fellowship work I did in Eric's lab, funded by the Dup15q Alliance, that also really matches well the technology that QState has developed. As I mentioned, going from sort of cell to cell to this, what we call our bright system, which we sort of incorporate all these different measurements of cells including using these movies and fluorescence to measure many hundreds of thousands at once, that's very well suited for understanding a disorder like Dup15q. So I think, at least for us, it's a combination of those few things. We really think we can get at the cause of Dup15q and sort of provide some therapeutic benefit. But yeah, I think like with Angelman syndrome, even for other people out there, that Dup15q is becoming more attractive just because it's on the radar. People know a bit more about it. So I think that's a little bit about how we got involved there. I do want to sort of mention that the therapeutic we're developing is an antisense oligonucleotide, which has now been going into patients for other disorders. So this is something, technology that's being used for lots of diseases. And what's really unique there is that it's a targeted genetic treatment. And so you can develop this, what we call, it's an ASO, that's the acronym. You can develop one that's specific for a gene. So in this case, UB3, which I think a lot of people are familiar with. And so rather than giving a small molecule drug, which has some off-target effects, and it's unclear maybe in some cases how it's benefiting people, this is a very targeted genetic therapy. So I think there's a variety of reasons to be excited and sort of work on Dup15q, but I think the technology at QState is uniquely suited to this disorder.
That's amazing. That's amazing. I definitely feel like this has to give hope to our families. So thank you. Thank you for, really focusing in on this disease and really trying to say, find the next future for therapeutics. And we're so fortunate that you have the history you do with Dup15q, and looking forward to seeing you at the science conference and just continuing the conversation. And I know you taught me already so much from things I've watched previously and then today as well. I'm literally taking notes on all of our calls today because I'm getting so much out of this. So thank you so much. And we look forward to just continuing to work with you.
Yeah, no, thank you for having me today. And, just to the entire Alliance, thank you. You don't need to thank me. I feel very, again, fortunate, and this group is extremely special, not just to me. Every time I go there, I just feel the energy. It's a really dedicated, passionate group of people, and really kind, friendly people. And it inspires me every day to sort of meet you all and to work on Dup15q. So thank you all.
Thank you. Thank you so much. And for everyone else, we will be back in just a few minutes with a couple of our Dup15q moms that you will not want to miss. So thank you.